All right, so we're going to take a parent that looks like this, and we're going to cross it with a parent that looks like this. So this is red axial, white terminal, and our F1 is going to be pink axial. So this is incomplete dominance because the heterozygous looks like a blend of phenotypes. It's not codominance. If it was codominance, the F1 would have splotches of red and white on the petals. But instead, it looks like the red and the white are blending together to create a pink phenotype. That's incomplete dominance. So we're going to cross with another heterozygous genotype. Now, normally this would produce a 9331. But we have to separate the homozygous dominant phenotype from the heterozygous phenotype for flower color. So it's going to look like a 9331, but it's not a 9331 because we have to pluck out these from what would normally be sort of inserted in the 9 and the 3. So I'm going to do Punnett squares. And now I'm just going to think of all the possible phenotypes. I could have red, yes, 1/4 chance. Can I have pink? Yes, 2/4 chance. Can I have white? Yes, 1/4 chance. Now for red, can I have axial? Yep, 3/4 chance. Can I have terminal? Yes, 1/4 chance, right? And the same applies to pink. Pink axial, pink terminal. White axial, white terminal. So the probability of each phenotype is just, we're using the multiplication rule. So the probability of making a red and axial is 1/4 times 3/4. Red terminal is 1/4 times 1/4. Pink axial is 2/4 times 3/4. You could write 1/2 here instead. We'll get the same proportion. Pink terminal is 2/4 times 1/4. White axial is 1/4 times 3/4. And white terminal is 1/4 times 1/4. All right, for number 2, we've got an organism that's going to self-pollinate. So you're going to cross it with the same genotype. Homozygous for all 3 dominant traits. So if you need to do a Punnett square off to the side, I'm just going to do it for A because it's going to look the same for T and R because we're crossing heterozygous with heterozygous for all letters. So homozygous for all 3 traits, 1/4 times 1/4 times 1/4, right? It's just this for A and T and R. For B, homozygous for all 3 recessive traits, again, it's 1/4 times 1/4 times 1/4. For C, heterozygous for all 3 characters, it's 2/4 times 2/4 times 2/4. And if you want to write it reduced as like 1/2 times 1/2 times 1/2, that's fine. Proportions are the same. Homozygous for axial and tall. So you can be homozygous this way or this way. But since they're saying axial and tall, those are the dominant phenotypes. So it's homozygous, 1/4 for axial and for tall. And then heterozygous for seed shape, that's going to be a 2/4 chance for my R crosses. All right, for number 3, the first parent must be big B, big B, or it's highly likely that the first parent is big B, big B, because they make 12 kids and all of the pigs are black. If this parent was big B, little b, I would expect approximately 50% to be albino, but I don't see that. The likelihood that this first parent is big B, little b, and that all 12 children have black fur is 1/2 to the power of 12. Right, so that's like tossing a coin and getting 12 heads in a row. Possible? Highly unlikely. So this is the better argument, that the first parent is big B, big B. And then when you take this albino and you cross it with another black pig, this time you get 7 black pigs and 5 albino pigs. So this looks like this is a black pig who is big B, little b, heterozygous. Right, so the first parent is true breeding. The second parent is not true breeding. Okay, so for number 4, capital P is a 1-pod dominant, lowercase p is a 3-pod recessive condition, capital L is normal leaf, and lowercase l is wrinkled. So just like I ask you to do Punnett squares one trait at a time, it's easiest to analyze one trait at a time. So let's just look at the pod. 1 pod. Everyone is 1 pod for A. So what's one possible genotype from the parents that would produce everyone having 1 pod? Well, you can make one parent homozygous dominant, and then you can make the other parent whatever you want. Doesn't matter. You can make it little p. You could make the parent big P, little p. You could make the parent big P, big P. Doesn't matter. Like, as long as one parent is homozygous dominant, the babies are all going to show the dominant trait. Normal leaf. It's a 3-to-1 ratio, normal to wrinkled, right? You've got 300 normal compared to about 100 wrinkled. So a 3-to-1 ratio, normal, wrinkled. What gives us a 3-to-1 ratio? Heterozygous with heterozygous. All right, B. You've got everyone shows the 3-pod condition. 3-pod is recessive. So if all your babies are showing recessive, the parents are going to have to be both homozygous recessive. And then you've got 300 normal leaf to 100 wrinkled leaf. That's the 3-to-1 ratio showing up yet again. So heterozygous for leaf. For C, all right, this looks crazy, but it's not bad. 1 trait at a time. 151 pod, 151 pod. That's about 300 1 pod. 53 pod, 53 pod. So approximately 100 3-pod. It's just a 3-to-1. Right, so this is the dominant. This is the recessive. So to get my 3-to-1, it's heterozygous, crossed with the heterozygous. Now let's look at the leaf condition. Let's do normal first, because normal is dominant. 150 normal, 50 normal, right? So that's like 200 normal. 150 wrinkled, 50 wrinkled, approximately. So that's 200 wrinkled. What cross gives me a 1-to-1? Well, it's not big L, big L, little l. That gives me a 3-to-1. What if I do big L with little l, right? So if I run that Punnett square, I get this, 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 this. Gives me a 1-to-1, which is what I see. So I'll make one parent heterozygous. I'll make the other parent homozygous recessive. D. That was D. What did I just do? Did I just get like halfway through C and then skip over to D? Did I skip C completely? Oh my gosh. What am I doing? All right, I just worked out D. I don't know what I did. But we said heterozygous for pod, and then heterozygous for one parent, homozygous for the other. Did I just straight up skip C? I don't know. 1 pod. Everybody has 1 pod. So again, 1 pod's dominant. So I'll make both parents homozygous dominant just because I can. Normal leaf, and they have normal leaf. Oh man, I'm feeling dangerous. So I'm going to make one homozygous dominant and the other homozygous recessive. Okay, C was so easy. That's why I skipped it, I guess. I don't know. Oh gosh. 1 pod. 1 pod. And that's it. Okay. So that's about 300. This is getting kind of messy. Hang on. It's about 300 one pod, so capital P. And here's a 3 pod. Here's a 3 pod. That's like 100 little p. All right, so it's a 3-to-1 ratio of dominant to recessive. So big P, little p, big P, little p. And now let's do leaves. Normal, normal. So that's like 220, 76. That's about 300. Normal, capital L, that's dominant. And then wrinkled is 72. Wrinkled, 27, that's about 100. Lowercase l. So it's a 3-to-1 dominant recessive. That's heterozygous crossed with a heterozygous. And look, this cross should produce a 9331 if the genes are on different chromosomes. That's a 9331. All right, I'm going to stop the video here just in case I crash. I don't want to lose all this progress. Check out the other video for more problems.